Hello, my name is Jeff Lester. I'm a medical doctor trained in internal medicine and medical genetics. I'm currently a practicing physician in Maryland. Let's get started. So I've created a series of lectures on YouTube to highlight to the general public the importance of genetics in their lives and explain how whole genome sequencing is going to change our lives and why people should learn more about it. The science is moving very fast and a lot of exciting things are happening. Topics we'll discuss include some basics of genetics, a brief on technologies in genetics, single gene disorders and complex disorders, genetics in the environment, cancer and the Cancer Genome Atlas, non-protein coding RNAs, GWAS and exon sequencing, the Human Genome Project, and the concept of the $1,000 genome. You can find out a lot more about all this geared towards the lay public and students by going to the National Institutes of Health website at www.genome.gov/students, or by becoming a member of my Facebook group, the $1,000 Genome Group, which I have recently started. There I discuss more advanced topics like epigenetics and the HapMap project. So again, this is designed to create genetic literacy and emphasize how important whole genome sequencing is and how it will impact our lives, and also a little bit about how medicine and genetics will change in the next 5 to 10 years. This is to emphasize that nucleus is inside our cells and that different structures with DNA in the nucleus create proteins. This is to emphasize that the DNA letters, which are shown there by the different colors of the pegs in number 1, form together to make genes number 3, and that genes together make chromosomes, shown in number 4, that chromosomes are inside the nucleus of our cells, which is shown in number 5, and that the cells obviously are inside our bodies. This is going from small to big. From biology, we know that there is DNA inside the nucleus of our cells, and this DNA makes up our genes. Here we see what DNA looks like. The 4 letters you get with DNA are A for adenine, C for cytosine, G for guanine, and then T for thymine. They fit together a certain way, and when you have lots of them linked together, you get the DNA ladder that we refer to as a double helix. Important to note that DNA letters are also sometimes called bases, and that bases are also sometimes called nucleotides. So when you're reading about genetics, you can see that these terms are used interchangeably. And in that small print there, you see a bunch of A's, C's, T's, and G's together. So picture pages and pages of these A's, C's, T's, and G's, and this is what genes look like when physicians and scientists sequence DNA. This is to address the basic tenet of genetics. We know that when we have a gene, the DNA that makes up the gene is double-stranded, and with the help of different proteins, the DNA in the gene undergoes a process called transcription to RNA, which is single-stranded. RNA then leaves the nucleus to the cytoplasm, which is just outside the nucleus. With the help of more proteins, undergoes a process called translation, and the protein coded for by the gene. Proteins are sometimes called enzymes. Proteins perform most of the functions in our cells to keep us healthy. Some proteins act as enzymes, which are large molecules that drive chemical reactions. Actually, the DNA letters code for different amino acids, but these amino acids are also joined up in strings in the cytoplasm, which make up the proteins. So a protein is basically a bunch of amino acids strung together like beads in a string. Depending on the types of amino acids that join together, they will start folding together in different configurations to make proteins and enzymes with different properties. The idea of the DNA of a gene coding for RNA and then protein is the basic premise of genetics. This slide is also to highlight that A plus B with an enzyme creates C in the body. But A plus B with a bad enzyme, you get no reaction, and with that you get a disease. So genetic diseases are commonly caused by errors in DNA, which leads to an error in RNA, which leads to a protein that does not work. On the bottom right, you have what a protein might look like after it's fully formed and ready for action. So the structure of DNA was first discovered by Francis Crick and James Watson at the University of Oxford in England in 1953. Their big discovery was given by one paper, which is 1 page long. Here it is. It goes to show that when you get to the real truth of something, it can be very simple. James Watson is alive and well. Actually, there's a conference that he's speaking up at Harvard in April, which I am attending, which I'm very excited about. In the 1960s, scientists learned more about chromosomes in human beings and their banding patterns and how karyotypes in certain patients have chromosome problems. Here you see a karyotype, which is a snapshot of chromosomes when they are very tightly packed. Actually, this is my karyotype, which I performed while I was in medical school. So, females have an XX, which are the last 2 chromosomes on the lower right of the karyotype, and males have an XY. There are 23 chromosomes in sperm. The last chromosome is either an X or a Y. And there are 23 chromosomes in egg, but egg had the last chromosome is an X only. When you have an egg and a sperm joining together, obviously you get 46 chromosomes. In the sperm you have 3.1 billion letters of DNA. In the egg you have 3.1 billion letters of DNA. Together they are 6.2 billion letters of DNA, which is the entire genome. And these are all the instructions needed to create an entire human. And this is what they addressed with the Human Genome Project, which we'll talk more about. They basically sequenced one complete set of DNA for 23 chromosomes. So in essence, they did half of the total instructions needed to make a human being. So once the chromosomes have been stained, scientists can look at the different banding patterns and look for extra or missing pieces of chromosomes, which obviously would include big chunks of DNA. So from small to big, we have DNA strands which make up genes, and hundreds of genes with non-coding DNA make up chromosomes. And the chromosomes are inside the nucleus of our cells and can be seen using microscopes. Obviously, there's a limit to the degree of resolution that the human eye can see down. The estimate is that geneticists can reliably see down to DNA segments smaller. Actually, geneticists cannot reliably see down to DNA segments smaller than 5 megabases in size, which would be 5 million letters of DNA. There have been cases on karyotypes where DNA segments as large as 20 megabases have been missed. So my point here is that the karyotype can give you a big picture of what the chromosomes look like. It is useful for diagnosing some diseases like Down syndrome, but many diseases need a closer look at the genetics to diagnose. So again, scientists can see down to 5 megabases at times, but not smaller reliably. The top karyotype on this slide is of a female. The one below is a boy with Down syndrome. He has 3 chromosome 21s, which is why Down syndrome is sometimes called trisomy 21. So we'll stop here and then we'll continue with the next lecture on the next slide.